Okay, I'd just like to begin by acknowledging that today we are on the unceded land of the Wurundjeri people of the Kulin Nation, and pay my respects to the elders past and present, and recognise that we have much to learn from the oldest living generations of scientists, teachers, artists and engineers who used and cared for the land on which WEHI operates at Kew, Bundoora, Parkville and Elizabeth Street. So it's a great pleasure for me today to present Aysha and Jiyi for the Wednesday seminar on behalf of all their supervisors. So we have Aysha, and who is a gastroenterologist, clinician scientist at the RMH and WEHI, and he's supervised by James Murphy, Andre Sampson and Britt Christensen at RMH. And then Jiyi on the other side, a basic scientist, supervised by myself and Kate Lawler and Sandra Nicholson. And this represents Aysha's PhD completion seminar, while Jiyi has just recently submitted her thesis. This is an enormous undertaking, this title here, just trying to decipher how cell death is dysregulated in inflammatory bowel disease. It's been, I think, combined, Jiyi and Aysha have spent maybe more than 8 years of work on this study, pretty much their entire PhDs, and it hasn't been without hardship. I think you'll see there's a preprint coming up on, well, there's a preprint on bioRxiv, but recent reviews, we had five reviewers, and you can imagine the number of additional work that was requested. So I thought just to make them feel a bit better, I'd start with a joke. So reflecting their official capacities as a clinician scientist and a basic scientist. So what's the difference between a basic scientist and a clinician scientist? One tortures mice to save humans. The other tortures humans with data from mice. That was generated by AI, by the way. That was not me. So, it's good to reflect for the younger people in the audience that studying cell death wouldn't have been possible without work in worms, where the cell death pathway conserved with humans was first identified and characterised. And also the key inhibitors of cell death, the inhibitor of apoptosis proteins, which are somewhat relevant to this project too, were first characterised in obscure viruses that infect moths. And perhaps in today's climate, the only person willing to fund that work would have been Doug Hilton. I'd just like to, I won't speak for too long, but I think it's good to say that Aysha and Jiyi have really led this work by themselves, and they've had a lot of, probably from myself, bad ideas. There was one day when I suggested some experiment probably requiring a huge amount of intestinal tissue, and Jiyi and Aysha kindly pointed out very politely that this would be akin to sending someone to Mars. They indicated that one biopsy from a patient is actually about the size of a grain of rice. So I think from today's seminar, you'll all agree with me that it's amazing what you can do with a few grains of rice. So I'd like to let Jiyi and Aysha take the stage. Thanks.
That is so nice, James, just the most beautiful introduction. And I would like to add that we certainly were the face of it, but this has just been a huge project by a lot of people, and a lot of people predating this project. So I would like to highlight that. And the preprint, as James alluded, is available now if you want to have a read in more detail. If you don't take away the findings from this project, I would take away that this has just been an eye-opening collaboration, and I would really encourage people to think outside the box and think about pursuing this kind of a project. If you can really just, take a leap of faith and hopefully come up with something that is akin to this project. And the number of people on this slide is probably falling short, and the people that I've highlighted there are people who have been really key players in leading this. So all of these people have led it together, to be honest with you. I would like to say special thank you to my supervisors, James, Andre and Britt, who have really been at the helm of this project, and obviously Jiyi supervisors. I would also like to say to Kamal, who's not here right now, and Sam, who they've done all the blots alongside Andre, because you'll find out that there were at least 1,100 of them, and Shanae and all of these guys, and Wayne, who have been on call to me at random times of the day, if I have a question about even something as simple as where is this reagent or how do I pipette, which unfortunately I still cannot do. Also, I would like to say thank you to Rory for coming on board this project. It wasn't something that they'd initially been aware of, and Mary Lou for taking a leap of faith as well, and obviously all these other people, James, our consumer, Bruce, obviously. I would like to say with James and Andre's group, they are literally the leaders of cell death, so I do feel incredibly privileged to have been included, and kind of, it's probably not something we could replicate. And with Britt, who has just, her excitement and passion about IBD is something that has no bounds, is highly infectious, so I'd say just beware. You will end up doing PhDs, all of us. So thank you to everybody, and obviously our family and friends who've been our supports behind the scene. So I'd just like to introduce IBD to begin with, and then I'll launch into necroptotic signalling from my side, and then hand over to Jiyi, who will be talking about unveiling mechanisms of apoptosis in IBD. So inflammatory bowel disease is a really complex chronic gastrointestinal condition that broadly encompasses ulcerative colitis and Crohn's disease, and they are the main two that I'll be talking about today. I won't go through the classifications of those, but just to clarify, ulcerative colitis affects only the large bowel. Crohn's disease affects anywhere along the gastrointestinal tract and tends to be more patchy. Ulcerative colitis can be just confined to the rectum. I'll just use my pointer. Can be confined to just the rectum, involve the left side or the whole colon, and Crohn's disease can be stricturing in its nature, so not just inflammatory, forming tight narrowings, causing bowel obstruction, or it can be fistulating, where it forms communications with intra-abdominal and intrapelvic organs. Both diseases can sometimes be accompanied by extraluminal manifestations, so you can get inflammation of the eyes, joints, or the hepatobiliary system as well. So the takeaway is really from IBD is that it is progressive. The inflammation can progress and result in stricture formation, which are the narrowings I talked about in Crohn's disease, and eventually lead to the requirement for surgery. And in some cases, if inflammation is uncontrolled or that chronic, you can end up with complications such as cancer. And what we're learning is that the deeper the level of remission or healing, the more beneficial it is for patients. So it is no longer sufficient to just focus on symptoms, but also focusing on biochemical markers such as circulating C-reactive protein or fecal calprotectin, and making sure that there's endoscopic healing at the mucosal level as well. More importantly, histologic remission or healing or lack of active inflammation is considered to be an aspirational goal, but we know that that's the best thing to achieve the best outcomes. IBD has a rising prevalence globally, and Australia is leading the pack. There are approximately 180,000 Australians who have IBD, and half of them still have active disease. And this is important because we have a lot of treatments that are in circulation at the moment, but I think we've reached a therapeutic ceiling. Interestingly as well, to note is that the paediatric population is only a small fraction of that, whereas the predominant inflammatory bowel disease we see is actually adult onset. This is important because a lot of the models that we use in the lab are actually more, depict more pediatric or very early onset monogenic cases, whereas in actual fact it's a much more heterogeneous disease, difficult to capture. There are advanced treatment options available, and I'm just going to highlight a few of the options that we have now. The reason I want to show you is because we do have targeted advanced therapies, and yet still we have a lack of treatment response to what we would like. So there's your anti-tumor necrosis factors such as infliximab or adalimumab. We have JAK inhibitors including upadacitinib and tofa. And we have anti-integrins that can also be used, such as vedolizumab, and these stop the sequestration of lymphocytes into the gut. There are anti-IL-12 and 23 agents in circulation at the moment, and we also have S1P receptor modulator agents which stop the egress of lymphocytes from lymphoid organs to reduce inflammation. And yet, despite this, 1 in 3 patients, if not 1 in 2, continue to need medical support, so they are refractory to current treatment. 1 in 4 will end up in hospital, and still about 25% of patients with Crohn's will need surgery at some point, and about 10% will need surgery with removal of a colon for ulcerative colitis. So it's clear there are other pathways at play, and I will introduce this slide, but I will let Jiyi speak about apoptosis, but it is busy, so I'll just talk you through it. Apoptosis and necroptosis are programmed cell death pathways, and the important thing to note is that when we're reproducing necroptosis in the lab, we are usually inhibiting apoptosis. So upon activation by, for example, tumour necrosis factor or pathogen response with Z-nucleic acid to ZBP1, we end up with an activation of the core signalling axis that I'll highlight here. So we've got RIPK1 activation and oligomerization that ends up forming a necrosome and activating RIPK3. These then recruit the executor, MLKL, which also in a sequence of activation events, ends up forming these little pores in the membrane and causes lysis. The important thing about this from a clinical perspective is that there are multiple clinical trials that are actually targeting RIPK1 in inflammatory bowel disease, and one of the phase 2 studies that has been published has actually failed to show efficacy. So we need to go back to the drawing board for this project, and really the reality is very few clinical cohorts have actually assessed cell death and inflammatory bowel disease, as we'll show you. And more importantly, so I guess some of the key findings were in 2011 is that caspase-8 deletion from intestinal epithelial cells did end up leading to necroptosis. So again, it's that concept of to demonstrate that there's potential for necroptotic signalling in IBD, but this is done on 4 patients, if even that. The Pia Domenico paper showed it in about 30 patients with IBD, but it was in patients who were children, and a later extension of that, and potentially in the same cohort, signified that there might be increased phospho-MLKL and RIPK3 in this population. However, what's important to note is a lot of these were done predating validated techniques. So in 2021, Andre and James and the lab validated a toolbox for immunofluorescence detection of RIPK1, 3, and MLKL as the core signaling antibody. And you could see here that they tested wild-type, untreated cells fixed in methanol using a WEHI-derived antibody or WEHI-created antibody by the group, and then in necroptotic conditions as well, and then looked at it against a gene knockout as a control. And you can see that really that at the forefront of cell death research. Subsequent to that, as part of the PhD, we also did established automated immunostaining protocols for allowing clinical translation of cell death detection in inflammatory bowel disease or in human tissue. And this is an example of some of the variables. This is actually a mouse example in mouse spleen, but basically we took a whole about 17 antibodies that are available for detection of the various core cell death proteins and optimize them against 21 using different about 21 different variables. So I think we tested about 300 conditions in the end to demonstrate that that there's about a 3-fold magnitude when you optimize these conditions for antibodies. And then when measured up in terms of quantification with mass spec, these lined up. And we later then applied them to treated HT29 cells under different conditions to demonstrate that they were in fact necrosomes, for example, in this necroptotic example. So the idea really is that predating this, we did not have good antibodies or detection methods that would be clinically translatable. And so we then embarked on this study, which is the largest cohort of adult IBD to be studied in this way. And this was a collaboration, obviously, with Royal Melbourne Hospital and the laboratory analysis done here at WEHI. We recruited 80 patients from the Royal Melbourne Hospital during their time of endoscopy. So patients who did not have inflammation at the time were excluded based on a specific criteria. They had to not have concurrent cancer at the time. And we took biopsies from 28 healthy controls. Then we recruited 25 patients with Crohn's and 27 patients with ulcerative colitis. We took biopsies from non-inflamed margin and inflamed segments from patients who had IBD, and then in patients who didn't have IBD, we just took the non-inflamed. So in total, we had more than 1,200 biopsies, 900 of which we've used for this project, and the rest we've banked. And then we've correlated this with their clinical data and sent some for histology, immunoblots, and then we did RNA sequencing for also microbiome analysis and host transcriptome, and then some underwent epithelial organoid assays. And so we derived, there is a lot of data, and we use this. We put a lot of the data into REDCap, and in coordination with the bioinformatician with us, Sai, big thanks to Sai for helping us, managed to do multiparametric correlation between these two. And so to begin with, this is what it looks like when we were collecting all the biopsies. So we took from every relevant site, including the small intestine and the large bowel, all the segments as well. And then depending on whether the patient had active inflammation, it was very easy to then say margin inflamed, non-inflamed. But where a lot of the patients were actually in remission or looked like they were in healing, we took biopsies from areas of historic disease involvement. And in our cohort, we've captured a lot of patients on lots of different therapies, including multimodal therapies. And I want to say thank you to James Rickard also for doing all the scoring, which is not standard at the moment for IBD, but basically using the Robarts Histopathology Index, he scored every single biopsy in a blinded manner, so he was not aware if they were IBD or control biopsies. He was only aware of the location for the purposes of the study. And what you can find is that these biopsies basically make sense and they are of appropriate quality. All the non-IBD, so all the little triangles represent patients with biopsies, So the patients that didn't have IBD, their biopsies were correctly at 0, and those that had non-inflamed were also basically at 0 or less than 3, which is in histologic remission. And then patients where we took biopsies from the margin looked like it was different again to the very inflamed or historically inflamed segment. So I think actually, I should add that of the biopsies, around 80% were completely in remission, so had a histology score of 3 or less. And this is remarkable because despite that, when we look at the signatures on these patients, it still looks like they are inflamed. And this is data from our cohort, but using the top 50 inflammation-associated genes from a published gene set. And we see that when we go from non-inflamed margin to inflamed relative to patients who don't have IBD, that these gradations are appropriate. The S100, A8 and A9 are code for transcripts for calpro, fecal calprotectin, and so again, that makes sense. When we look at the gene set enrichment analysis on the right here, you can see that appropriately as well, and as expected, TNF signaling and interferon pathways are dysregulated, and it looks like apoptosis here too. Oh, is that... I was expecting a rubber chicken to come up today, but what's really important is despite this dysregulated cell death is not a histologic hallmark of IBD clinically, nor does it feature in the histologic algorithm. And what you can see, and again, I'm not a pathologist, to be honest, this is, again, thank you to James Rickard for highlighting this, but what you can see here is 3 examples of patients with IBD up the top. So we have a patient with Crohn's disease who's really well controlled based on a Robarts Histology Index of 0, with next to no appreciable evidence of cell death. This is a patient with Crohn's disease who's on an anti-TNF and immunomodulator and steroids because they were having a flare. Their Robarts indicated a histology index that was severe at 20 and only potentially 1 So similar, one apoptotic body, clinically appreciable potentially. And when we look at a patient with ulcerative colitis who's also in histologic remission in this particular case, they also don't have any significant features. So it's really remarkable because when you look at, for example, a different disease entity where apoptotic bodies or apoptosis is considered clinically significant, such as GVHD or graft versus host, you can see that apoptosis is very clinically apparent. And so I'll take you through a couple of the examples that we have. This is a patient with ulcerative colitis. So on the left, I've got the core signaling axis for necroptotic signaling, just as a marker, a reminder of why these are important and that this is the sequence that is previously thought. And this is a panel of one example of a profile of a patient. But like I said, we've done about 1,100. Well, not me, Sam and Kamal and Andre. And we've actually used cell controls, and we've used an IBD patient loaded with biopsies from a control patient just to optimize how we interpret this. And so what you can see here, and I've also demonstrated here that this patient's endoscopy looks completely normal. You can see down the bottom, the Robarts histopathology index. Again, all the biopsies are completely considered histologically normal. So when we look at this, we'll start from the top. You can see that there is activation of phospho-MLKL in this case, and that there is activation of RIPK3, but that this is not actually happening in the presence of activated phospho-RIP1, which is an upstream marker and thought to be necessary for this to occur. More interestingly, you see ZPP1 activation in this sample. There is still also presence of caspases, although they're not being activated here. And so this is important because it raises the question of, well, what is signaling the necroptotic pathway in here if it's not RIPK1? And this, I guess, challenges the RIPK1 inhibitor trials. What we see in a different example in a patient who does have inflammation down the bottom here in their inflamed sample. So this is a patient with Crohn's disease on an immunomodulator, and you can see that their disease looks endoscopically apparently active, as it is. This is from that patient on that day, that we see that there is, no real convincing phospho-MLKL, but you can see that there is activation of activation of RIP3 as signified by phospho-RIPK3, and that there is ZPP1 breakdown, but again, not happening in the presence of active RIPK1. And in fact, you can see cleavage of some apoptotic specific enzymes, so caspase 10, 8, and 3, all being activated together. And so it suggests that RIPK3 is an early active event, followed by necroptosis and inflammation. And this is a heat map, which summarizes all of our data pooled from the immunoblots, which we actually quantified. So we quantified all of those little barcodes. Yes, we actually did. And then we put them and we quantified them and then analyzed them in this heat map. And you can see that in this particular example of phospho-RIP3, so we have plotted all the quants for this patient, for this protein, looking at each biopsy is actually, each dot is a biopsy, and you can see a statistically significant increase with inflammation. I'll just bring your attention again that phospho-RIP3 appears to be the earliest change that is statistically significant from non-inflamed to inflamed, and that this is occurring in concurrence with cleaved caspase 10 and 3 as inflammation increases. This is also occurring despite the, no statistical significant loss of CIAP, although there is a trend to go down. And so we are posing this idea of necroptotic to apoptotic signaling in IBD, which challenges our understanding of cell death. And I'll just show you this heat map of bulk RNA sequencing, which summarizes 164 necroptotic and apoptotic genes, which when we put which we analyzed with unsupervised clustering, actually came up with only a few gene transcripts that were statistically significant with inflammation, and they happen to cluster together and be co-regulated together by NF-kappa B and interferon responses, and I'll let Jiyi go into that in more detail. But this is interesting because it suggests only a small number of genes need to be modified for to have such a huge effect in inflammation at the protein level. When we looked at the cell type deconvolution using cybersort, we also see that there wasn't a major inflammation-dependent difference in immune cell infiltration relative to controls. So it does suggest potentially changes at the epithelial level, and I'll let Jiyi go into that in more detail. And ultimately, it's probably because our patients were all in remission at the time, predominantly. And so I'll summarise before passing over to Jiyi, but basically necroptotic signalling does appear to arise in inflammatory bowel disease, even in patients in histologic remission that we would otherwise consider to be in the best medical management as possible, and this is signified by phospho-RIPK3 is RIPK dependent in parts and can be activated by suggests ZBP1 correlation as well. There is this concept also that there could be sublethal necroptosis because we don't see activation of the necroptotic executioner MLK or cell death in inflammatory bowel disease. And I will pass on to Jiyi, who will discuss more about the apoptotic signaling, which we also see elevated.
Thank you. Oh, there's no laser. Okay. All right. Then maybe I adjust and then that coming out. Yes, cool. All right. Thanks, Aysha. So I'm going to take over and go into the part 3 of our talk, which is mainly about the cell death mechanisms in IBD. So, as Aysha just discussed before, the key findings we found from the IBD patient biopsies is upregulation of cell death pathway. But since biopsies is a mixture of different cell types, so which cells are most affected by cell death is the first question we want to have a look. So to investigate this, we reanalyzed a publicly available IBD single-cell RNA sequencing data, and we assessed the apoptosis pathway enrichment across the different intestinal epithelium cells. So you can see here, the different types of cell were separated by different color, including epithelial cells and immune cells. But if we jump into to look at the pathway enrichment, we could say this group of cells, which mainly is epithelial cells, are more enriched in apoptosis compared in immune cells. And similarly, when we perform the immunohistochemistry of cleaved kase 3, which is a marker of apoptosis on the non-IBD and IBD biopsies, we could only see the positive signaling mainly pop out in the epithelia, but not in the immune cells. So this reinforces that the apoptosis is specifically enriched in the intestinal epithelium of IBD patients. And moreover, if we take a closer look of this result, we could see this group of cells, which mainly contains clonocytes, cryptop clonocytes, and undifferentiated A cells, are specifically enriched in apoptosis. So this is quite interesting. What are these cells? So here's a model depicting, I know, very simple model, depicting the structure of small intestine. So it's composed by 3 main parts. In the middle is epithelium, which has a crypt and villi function. And then above the epithelium is the intestinal lumen, which has a mucus and microorganism inside of it. And below the epithelium, we call lamina propria, where all the immune cells were located in. And as we can see at the bottom of the crypt, there is a cell called undifferentiated cell, also known as epithelial stem cell. So they are required for the epithelium cell renewal and differentiation. And 80% of the rest of the epithelium cells are enterocytes or clonocytes. They are required for the nutrients absorptions. So these 2 types of cells plus some secretory cells, they formed a monolayer of epithelium, and that acts kind of like a barrier like chemical and physical barrier. It can segregate the microorganism and the immune cells. It can also sense the microbiome stimuli and trigger some appropriate immune regulation. And in the normal condition, every epithelial cells reach their end of life every 4 to 5 days when they reach the tip of the villi. However, if the cells is dying out of control, it might trigger the gut inflammation. So for example, if we got excessive colonocyte cell death, it might impair the barrier function of the epithelium. And if we lost the intestinal stem cell, it will dampen the epithelial repairment, and those two factors will trigger a leaky gut, which allows the microbiota to translocate into the lamina propria and activate those immune cells. Then those immune cells will release pro-inflammatory cytokines that triggers inflammation, and also those cytokines may also, again, further trigger the epithelial cell death, and that's kind of become like a vicious cycle. So now the question is, how can we study this model in vitro? So in this study, instead of using 2D colorectal cancer cell line, we choose to generate human intestinal organoids and use them as our in vitro model. It's because organoids, as a three-dimensional cultured model, can closely mimic the structure and function of human intestine. Epithelium especially, and in this study, we generated organoids from the stem cell of the healthy donors' biopsies, and then without differentiation, we can see they form like cyst-like spheroids. And then after differentiation, we can add different smaller molecules to trigger the differentiation, and the organoids can be differentiated into any types of epithelial cells. And after differentiation, they gain the crypt and villi structure, so which is pretty cool. And in our study, we specially choose a Wnt inhibitor called IWP2 and add this inhibitor into the organoids differentiation media to specifically get the colonocytes-enriched organoids. And here's the quality control experiment we performed. So here we include 4 donors' organoids, and then we generated them in undifferentiated situation, and then trigger the differentiation with or without IWP2. And then we extracted RNA and performed the qPCR to test some key genes. So for example, we can see the stem cell gene, LGR5, is significantly downregulated after differentiation, while the colonocyte marker LP and FABP1 is especially upregulated in IWP2-treated organoids compared to the normal differentiation organoids. So this tells us that we got the good strategy to get undifferentiated and colonocytes-enriched organoids. Now the question is, how can we use the organoids to mimic what we saw in the patients before the IBD cell death signaling? So to understand this, I need to bring this data back, which has been covered by Aysha. So this is GSEA analysis we performed on our patient's RNA-seq. And as we can see here, the TNF signaling, interferon gamma signaling, and the apoptosis signaling are significantly enriched in IBD samples. And more importantly, TNF and interferon gamma, they are the key pro-inflammatory cytokine in IBD disease because the drugs targeting them, such as anti-TNF and JAK inhibitor, which targets the downstream of interferon pathway, has been used clinically in the IBD treatment. And moreover, in our previous study, as well as another group, we found interferon gamma combined with TNF can cause mouse intestinal epithelial cell death and mouse macrophage death. But whether these 2 cytokines can also induce the human intestinal epithelial cell death remains unknown, and that triggers our hypothesis, which is interferon gamma and TNF can trigger the epithelial cell death, and that contributes to the pathogenesis of IBD. So the first question we want to test is whether these 2 cytokines can reproduce what we saw before in the IBD patients. So we tested the cell death gene expression at first. So again, this is the data we shown before, which represents the cell death gene expression of IBD patients compared to the non-IBD patients. And we performed the exactly same analysis on stem cell organoids and colonocyte organoids treated by interferon gamma, TNF, or gamma combined with TNF. And hopefully you can appreciate how similar they look to each other. So this tells us, yes, those cytokines kind of can reproduce the cell death gene expression, But how about cell death itself? So there are so many ways people have already found to measure the organoid's death, but none of them are accurate enough in our own hand. So with the help of Imaging Lab, we generated a new method to accurately measure the organoid's death by using the real-time imaging machine InQSight. So basically, we added the appropriate item, which is the PI stand for the dead cell, into the cell culture, and then we use a machine to take the time course pictures of the organoids. And then the proportion of the cell death was calculated by PI positive organoids area divided by total organoids area. So now, having this analyzed method, we had a look of interferon gamma and TNF treatment by itself in organoids at first. So here we can see after 21 to 24 hours, we didn't see interferon gamma or TNF itself trigger any significant cell death in both stem cell or colonocytes organoids. However, when we combined the interferon gamma and TNF together, the cell death killing is significantly increased. And I include 2 videos here showing the organoids are gradually killed by interferon gamma and TNF, especially in the colonocytes organoids. After 24 hours, almost they just all dead. Yeah. So this tells us so interferon gamma synergized with TNF to induce the intestinal organoid cell death, and this can be a robust in vitro model if we want to do some research on the epithelial cell death in the IBD tissue. Yeah. So of course the next question is now how is this cell death is happened? This is a graph showing the apoptosis. So as we mentioned, the programmed cell death can be classified into apoptosis, necroptosis, or pyroptosis, roughly. And apoptosis and pyroptosis, they are the caspase dependent cell deaths. So what is caspase? So caspase are the enzyme which can cut the substrate and trigger the cell death and the inflammation response. So first, for apoptosis, it can be divided by intrinsic apoptosis and extrinsic apoptosis. The intrinsic apoptosis is mitochondria dependent and back dependent, and it will trigger the caspase 9 activation, while the intrinsic apoptosis is dependent on caspase 8 and caspase 10. But both of the pathway will trigger caspase 3 and 7 activation, and in the end, trigger the apoptosis. So to test if this pathway is activated and plays some roles, we run a Western blot, including stem cell and colonocytes organoids. So let's use the colonocytes organoids and caspase 8 staining as example. So I treat them with interferon gamma by itself, TNF by itself, or interferon gamma combined with TNF with different time course. And as we can see here, the full length of caspase 8 is increased in interferon gamma treatment by itself, but it didn't trigger a lot of cleavage, while TNF by itself almost didn't do a lot of things. However, when we combine interferon gamma and TNF, we could see the full length of caspase 8 is significantly cleaved and become this P18 fragment, which is active form of caspase 8. And the same trend happened in caspase 9, caspase 10, and caspase 3, and they're all involved in apoptosis pathway. And if we look at the stem cell, the trend is also quite similar, but just less levels of the activation. So this telling us apoptosis is involved. Now, how about pyroptosis? The pyroptosis is an inflammatory cell death regulated by gasthamin D. So gasthamin D can be cleaved by caspase 1, caspase 4, and caspase 5. And when it be cleaved, it will move to the cell membrane and cause the cell pore and then induce the cell death. And in our Western blot, we also found caspase 1 is upregulated by interferon gamma and started to have a strong cleavage after gamma and TNF combination. So this indicates interferon gamma and TNF can induce a strong caspase cleavage. Now, how about necroptosis? So as Aysha already go through this pathway, let's jump into the Western blot directly. So as we all heard, the phosphoryb-K3, phospho-MLKL, and ZBP1 are the regulators of the necroptosis. And in my organoids, again, with interferon gamma by itself or interferon gamma combined with TNF, we could see the significant upregulation of these key markers. So these tell us necroptosis is also activated after interferon gamma and TNF treatment. So as a summary, now we got all those regulated cell death activation, but which one plays the main key roles? Or in another word, how can we stop this kind of death? So to identify this, we decided to use a series of inhibitors. So here, without any inhibitors, we can see interferon gamma and TNF triggered around 80% of colonocytes death after 24 hours. And we firstly added the GSK-A72, which is a necroptosis inhibitor, into the system, but it ends up with no difference of the cell death. So this tell us necroptosis is not playing the key roles in the interferon gamma and TNF killing. And the same trend happened when we use the pyroptosis inhibitor, MCC950 or VX675. So this telling us pyroptosis is also not the key regulators of this killing. And lastly, we choose to use pan-K-space inhibitor, QVD. And we performed an assay called DVDS assay to make sure that QVD is fully blocked the K-spaces. So here we can see this is a K-space 3 activity assay, and we can see after interferon gamma and TNF treatment, the K-space 3 activity is extremely high, but QVD can fully block it. However, when we jump back to the cell death assay, it's very strikingly, we still couldn't see the fully protection of cell death. Instead, QVD and QVD plus all those previous inhibitors can only delay the cell death. So who is still activating if we block all of this? We think it's because of mitochondrial apoptosis. Because the permeabilization of mitochondria can eventually cause apoptosis even in the absence of K-spaces. So to test this hypothesis, I overexpressed a pro-survival BCL2 protein, which is BCL2, in my organoids to stop the back-and-back activation. And for this Western blot, we can say, yes, this overexpression is successful. It's very high concentration. And finally, this time when we treated the Y-type and BCL2 overexpressed organoids with different time of gamma and TNF treatment, we could see a very nice protection even after 24 hours of treatment. So this tells us this interferon gamma and TNF-induced cell death is mainly a mitochondrial apoptosis via back-and-back. So as a quick summary of what I've shown so far, we found the necroptotic and apoptotic pathway are dysregulated in IBD intestinal epithelium, and using organoids, we found interferon gamma and TNF can induce the organoids' death, and this is a back-and-back dependent mitochondrial apoptosis. Then the next question I want to answer is, how is back-and-back activate then? So to understand let's jump back to the BCL2 family member. Because they are most relevant proteins to back-and-back. So there are pro-survival proteins and the pro-apoptotic proteins exist in this family. And firstly, I hypothesized the interferon gamma and TNF might regulate the pro-survival protein directly to activate back-and-back. But we need to know that these different pro-survival proteins, they are not exactly equivalent. Sometimes they are overlapping. Sometimes some of them may play the dominant roles. So by using these pro-survival inhibitors, different one, we found the BCL-XL inhibitor and maybe the MCL1 inhibitor can trigger the organoid cell death by itself, while the BCL2 inhibitor cannot. So this put BCL-XL and MCL1 as the possible candidate. However, when I run a Western blot and test the level of them, again, this is stem cell and colonocytes, we cannot see any of them significantly reduced after interferon gamma and TNF treatment. So if it's not the pro-survival protein, it might be because the pro-apoptotic protein, because they can react on those pro-survival protein and trigger back-and-back. So here I had a look of Puma, Noxa, and Beat, and interestingly, we found Puma is significantly induced after interferon gamma and TNF, especially in the stem cell compared to clonocytes, while the NOXA and the beads has almost the same level with all the treatment. So now the next question is, yes, we saw the PUMA upregulation, then does PUMA regulate the cell death? So by using CRISPR-Cas9, we successfully delete PUMA gene, which is BBC3, in both stem cell and clonocyte organoids. And very interestingly, in stem cell organoids, we could see deleting PUMA successfully blocked the interferon gamma and TNF interferon gamma and TNF triggered cell death. And here is the representative picture of the last time point, and here's the analysis data. However, when we move to the clonocytes, this protection is disappeared, which means PUMA is not required for the clonocytes organoids death. So as we found PUMA is involved in stem cell, we want to go back and to look at if our IBD patient samples also have the upregulated PUMA. And the answer is yes. So we compared 11 pairs of non-IBD and IBD patients, but we found like 4 pairs of them showed the upregulated PUMA in the IBD patients. And there's another group published before. They include more ulcerative colitis patients. They also found a proportion of the patients showed upregulated PUMA. We speculated it might be because the different way to take the biopsies, as you have to like dig very deep to get the stem cell. And most importantly, this group also trying to delete the PUMA in the mouse model, and they found deleting PUMA can alleviate DSAs, induce the colitis in mice. So they put PUMA as a possible candidate for the IBD treatment. Then how about the clonocytes? If it's not PUMA, what is that? The first things jumping out in our mind is iNOS, or inducible nitric oxide synthesis. Because we previously found in mouse macrophage, iNOS is required for interferon gamma plus TNF-induced mitochondrial cell death. What is iNOS? iNOS is an enzyme which catalyzes the production of nitric oxide from the L-arginine. And interestingly, in our own research, the gene of iNOS, which is NOS2, is one of the most upregulated genes in our IBD patients' RNA-seq. And the same trend happened when we reanalyzed the published database with 600 IBD patients, including both Crohn's disease and ulcerative colitis. And more importantly, when we moved from RNA level to the protein level, we performed the IHC staining of iNOS in the non-IBD and IBD inflamed tissue. And here's a representative picture. So we could see the non-inflamed patients, they barely have no like iNOS expression level. However, in the IBD non-inflamed and inflamed tissue, we could see iNOS expressed, especially in the epithelial part. So what is the iNOS is doing? So since 1990s, people have already started to mention they found the upregulated iNOS in IBD. But so far, we still don't know what is this protein is doing. And it's especially because studying iNOS in human is really difficult because, first of all, the mouse and human, they have a huge discrepancy on iNOS expression. For example, the mouse macrophage can express iNOS easily, while human monocytes cannot. And also the colorectal cancer-generated human cell line can also not express iNOS. However, using organoids, we found interferon gamma treatment and also interferon gamma combined with TNF treatment can significantly produce iNOS in both stem cell and clonocytes, especially in clonocytes. And we also measured the soluble level of iNOS, which is nitrite, and this also showed the same trends. So this high level of iNOS must be doing something. Yeah, and when we knock out iNOS, which is a NOS2 genes, and this is a very interesting result, because this time in the stem cell, when we deleting NOS2, we can see there's a good deletion efficiency because nitrite level is significantly down after gamma and TNF treatment, but it didn't stop the stem cell killing from the interferon gamma and TNF. However, in the clonocytes, even though the knockout efficiency is not very high, but we can see a significant protection, which you can also see from this representative picture from the interferon gamma and TNF treatment. So due to the time, I might jump to the summary directly. So in this study, we show that the excessive cell death signaling activation is an underlying feature of IBD, and potentially there's a necroptotic to apoptotic axis exist. And mechanismally, we found the necroptotic signaling represented by upregulated ZBP1 and phosphoryl K3 prominent in histologically non-inflamed IBD tissue. And we speculate this is a sublethal function and might acting as a trigger for inflammation signaling and immune activation. However, as inflammation intensified, apoptosis pathway will jump out and become the dominant, and that might trigger the tissue damage. And using interferon gamma treated organoids, interferon gamma and TNF treated organoids, we found that the cell death outcome is due to the back-stem-back activation and through mitochondria. And we found inos and puma as the two different regulators in two different types of cells. But we still don't know what is the exactly non-lethal function of the necroptotic pathway, and that requires further study. So in the end, I'd like to thank my PhD supervisors, James, Sandra, and Kate, and my committees, all the members from Vince Lab, Murphy Lab, and all the authors contribute to our current paper. And I think I will include Aysha in the QC question, and we're happy to answer any questions.
Hello?
Yep.
Hey, doesn't always work. Frequent flyers will know that. That was brilliant. Thank you both. I'm glad we got through it with heaps of time for questions. If anyone has any questions, please. Oh, I'll start with Andrew.
Congratulations to both of you and to all involved in some lovely work there. Correct me if I've misunderstood, but in the patient biopsies where the disease was healed while they were on therapy, there was still a signal of maybe some form frust of necroptosis. So have you been able to recapitulate that in the organoids at all by including some of the inhibitors that are used in therapy to recapitulate that? Because it just seems to me there's always a gap even in great stories, but just to help with the inference.
Yeah, we haven't. It's a very good point, and we definitely need to do more optimization on this organoid system, because if we want to study on the non-lethal function, which is necroptotic signaling, we might need to reduce the levels of the cytokines, because we don't want to kill them directly. So that's the things we're going to test in the future. Yeah.
Congratulations to the both of you for your work. I'm coming from a cancer background, and your findings are very interesting because in cancer usually try to induce cell death to prevent tumor growth. However, in IBD you do the opposite, which makes sense when you consider the leaky gut syndrome. I guess what I want to ask is, do you think that there's a risk that when you block cell death, that you might induce neoplastic lesions or dysplasia in your patients?
Yeah, exactly. So I think the key question we still cannot answer here is, even though we're looking at the cell death pathway, but we don't know the cell death is actually a good things or bad things in the IBD patients. So of course, we can use interferon gamma and TNF kills them and they look like dead, but maybe that is a protected way that the body used to protect from the cancer or like the generation, especially the key regulators inos we identify here. The mutations of inos people also find is like a gene risk for the colorectal cancer generation. So we just, don't know. Yeah.
I'm not seeing too many hands up. I'm going to ask you a question. Can you tell us a bit more about caspase 8 versus caspase 10? So what do you think's going on there?
Well, in my hand, they don't have a huge difference. They always kind of like good buddies, stay with each other and cleave together. But that's a really good point. I don't know. Yeah.
And echoing the comments before, my question is, when you looked at the patient samples, hypoxia seemed to be It was down the bottom, but it was really making a big change in the level of signaling, and it was particularly strong in the inflamed tissue. So I guess I'm wondering whether that is something to, what are your thoughts with regard to the organoid work on that? Because it really seemed to be quite strong. And I guess I would perhaps preempt my first question by saying, have you sort of confirmed that there is hypoxia with Western blots and anything like that?
Hypoxia.
Hypoxia is less of oxygen.
Yeah.
Yeah, that's
I know.
yeah, that's interesting, and especially nitroxide has some competent functions with oxygen. And if you have really high level of nitroxide for a long time, it might trigger the hypoxia. Yeah, but yeah, we haven't tested that before. Yeah, and yeah.
I had a question relating to the clinical samples. You had an overlay of apoptosis occurring in the colon stem cells, and that's what you focused on for a lot of the work. Do you see a shortening of the crypts in your patients who have no signs of IBD? Like, is there still a physical adaptation there? You got reduced colon crypt length due to the stem cells.
For a healthy patient?
So in the non-inflamed IBD patients.
Yep. Sorry, did you want me to show that? Oh, no, we haven't. No, we haven't, but it's a good thought. It's not usually associated with a healthy control, but a pathologist may have more, there are some subtle changes sometimes that we do see even in patients who are considered healthy, but we did measure all of those things actually qualitatively as well. So if I look back through different biopsies, I may have a different answer, but we definitely didn't measure the actual crypts.
In IBD patients who have active disease, you'll get crypt branching, so you actually get link in the IBD patient compared to control.
Yeah, but just not in controls. Yeah.
Where you don't see the scoring of IBD, you've got a molecular change and upregulation of apoptosis. So do you see that in those patient biopsies?
You can know the healed, so when the Roberts score does not take into consideration crypt branching, you just see that, so it does not measure normalisation. So it's something we could look at as a secondary outcome in IBD. You'll have more branching, you still have a normal Roberts score. Yeah.
We did actually put that into the additional descriptors after the RHI, but we didn't analyse the data based on those. So we do have that data. Yeah.
Hello, great talk. I'm super interested in understanding what triggers this. You talked about how the majority of individuals who develop IDB are older, I guess middle-aged individuals, and obviously apoptosis is really important in the natural flux of the gut. So what triggers this transition from natural apoptosis processes to becoming IBD in older individuals?
So I'll answer for one part, but then Jiyi will probably take over for the... So, it's adult onset, but actually the IBD prevalence occurs in patients who are of a peak reproductive and later adolescence. It's just, I was just trying to make the distinction that it's not the same to study, some of the gene knockout mice or gene mouse models that we have as a signifier purely of adult onset IBD. And the second point regarding the apoptotic signalling, I think we're seeing increased apoptotic signalling in the tissue based on the immunoblots and the RNA results. But I think the thing that we can't quite reconcile is that we're not seeing active apoptotic dying cells in the histology of these patients. We haven't obviously measured that, but in the predominant most of the biopsies, we're not seeing that. So we're calling it signalling in that regard, but when it's recapitulated in intestinal organoids, they are dying, but presumably because the cytokine doses are different. And yeah, in terms of the trigger, we're not sure. There's a lot of theories going around, environmental, viral, potentially something in the dysbiosis. We haven't analysed the data in detail with that, but it could be something to do with that. Yep.
Great, thank you both for that talk, and congrats to the whole team on the work. It's a great effort. I had a question around that sublethal necroptosis observation, and you can kind of imagine a situation where you could molecularly stratify patients based on that signature. And I wondered whether you could correlate from your clinical data, is there any link between the therapy lines those patients had received or were receiving and their emergence of that sublethal necroptotic signature?
Yeah, so on initial analysis, there didn't appear to be a difference between the different classes, but in all truth, I think we just haven't captured enough numbers. So we've got, we analysed patients at about, I think there were about 30 patients with IBD that we've analysed on a lot of these data that I've presented, but actually we haven't analysed by sub-treatment group, and I'm really interested in looking at the classifications of, say, Crohn's disease itself. I think we just need more numbers before we show that. We're very interested in that too.
It could actually be a molecular marker predicting relapse, so we want to be able to actually... Sorry, I always talk. I can't help myself. But I've got Aysha now doing the clinical outcomes to see whether actually this is a predictor of relapse, because that's the next holy grail is molecular healing, and is this going to be able to predict those patients who are actually going to relapse, and can we target that?
Thanks. We're out of time. I'm just going to ask everyone to join me in thanking our speakers. And thanks everyone for coming. Thanks for the great discussion.